Welcome to the second part of Best Practices for Hit Generation. In this webinar, we're going to start looking into classical screening strategies. And we call them classical because to date they're still the most widely used approaches for hit generation. So here we're going to look into screening approaches, and the following webinars will take care of the next step, which is hit validation, followed by looking into possible screening technologies. So here we're talking about really the first step of hit generation, which is the identification of hits. And if you remember the introduction, hits are defined as compounds which modulate the activity of your target of interest in a particular assay. So here there are two components to consider. One is the screening library. Which compounds are you going to screen? And the second one is the screening assay. Which assay are you going to use to monitor the activity of your target? Once you have these two, you can perform your screening, which will give you the hits. Now, one important thing to consider when you talk about selection of compounds is that it's based on the screening assay itself and the knowledge you have on your target. So we can separate or classify the classical approaches into three buckets. First, the unbiased screen, which is the high throughput screen of a large set of small molecules. In this case, the size of the library will depend on the complexity of the assay. The second bucket is the focus screen, where we're going to screening of a smaller set of compounds based on the previous knowledge we have of the target. And the third bucket, called Insidical screening, is going to use computational methods to select the compounds that are going to be screened experimentally. So let's go into the first approach, unbiased screen. This is probably to date still the most widely used approach for hit generation. And here we rely on screening a large number of compounds. The idea being the more compounds you screen, the higher the chance you have to find hits on your target of interest. So in order to do this, you have to have access to a compound library. And pharmaceutical companies and academic institutions have assembled these libraries by purchasing commercial compounds, designing custom libraries to increase the diversity, but also by collecting compounds that are made through their discovery projects. Last but not least, several collections also contain natural products. Now, since we're talking about screening a large number of compounds, it's important that we use robotics in order to automate the screening. And then you can use plates, such as the one you see in this picture here, where each well contains an individual compound in order to be able to screen all the ones you want to screen. After running your assay, you're going to get a lot of data that you're going to be analyzing in order to define your hits. Now, the selection of the compounds you will screen is based on the assay capacity and costs. Typically, you would screen about 250,000 compounds, but it may vary a lot, going from 50,000 for complex cellular assays to several millions if we talk about simple enzymatic assays, for example. And this selection is done using several criteria. Diversity, of course, is important. You have to have a diverse set of compounds if you want to find some that hit your target. You may want to exclude undesired properties such as low solubility or high lipophilicity. And you also may want to exclude frequent hitters, which are compounds that were found to hit many targets, or compounds containing Payne's motifs, pan-assay interference compounds, which are known to interfere with biological assay systems. Now, for a long time, the default was, if your assay allows it, you should screen the whole library. But this has changed over the years, and many people now think that screening a subset of the library not only will save you time and money, but also may be more successful. So institutions have approached this differently, and we would like to share the example of the Novartis screening deck, which has been published in 2020. So here, the approach is to maximize the diversity of the compounds that are screened with a bias for properties that are favorable for screening. So you organize your screening library into grids according to properties and diversity. And the process is explained in this cartoon here. First, the compounds are annotated. They can either have a positive flag, these are the dark blue ones, because the compounds have high solubility and high permeability. They can also have no flags, these are the light blue, or they can have a warning flag. These are compounds that may have reduced solubility, compounds that are known frequent hitters, that have undesired substructures, or samples of limited purity. The second step is to allocate the compounds into diversity classes. And diversity can be defined by chemical diversity, different scaffolds, 2D and 3D clustering, or by biological properties, meaning activity on targets or HTS fingerprints. So now you have your compounds that belong to one or several diversity classes, and they have these annotations based on properties. The third step is to pick the best compounds from each class. So looking into the example here, so first we're going to select compound A to cover class 1, and we're going to select it over compound B because it covers also class 3. If we look now at class 2, we're going to select compound C because it has the best properties among the compounds in this class. And then to cover class 4, we're going to select compound P because it also overlaps with class 2. And at each round, we're going to select the next best compound. The outcome of this is that you have a grid of compounds that are ranked in the columns according to their properties and in the rows according to their diversity. And the power of this is that you can really select any plate you want in this grid. Usually, of course, you start with the compounds with the best property, and the lower you go in the rows, the higher the coverage you have in your library. That way you can also easily adapt the number of compounds screened to your assay. Now, in addition to being able to select the compounds you want to screen, this has led to an increased use of the so-called iterative screening approach. The idea there is that you screen a small selection followed by screening nearest neighbors of the hits iteratively. And this has been shown to lead to superior results than screening large compound sets. It's important to mention that this is one example, and companies have taken different approaches based on their own learnings from historical screens. So now you've decided which compounds you're going to screen. But before actually performing the screen itself, it's important to test your assay in a pilot phase. The objective here is to use a small diversity set, maybe 5,000, 10,000 compounds, to test how your screening assay and setup will perform. So you want to assess the assay quality and robustness. And here we'd like to introduce an important measure called the Z prime factor, which looks at the separation between active or positive control and negative control. The idea behind this is that because every biological assay is variable, you want to assess the statistic validity of your assay, meaning the probability that a positive result comes from the variation of the inactive compounds. A Z prime over 0.5 is considered adequate, but of course you would aim higher, and 0.7 is considered excellent. Because your compounds are dissolved in the MSO, you want to look at the effect of the MSO on your assay. And finally, because your assay will run over several days, you want to assess reagent stability. You may want to try running your assay with different conditions, detergents, incubation time, in order to see which ones performs the best. And you want to get a first idea of your hit rate. How many compounds are actually going to be found when you run the full screen? If it's too high, then your assay may be too sensitive. If it's too low, maybe you're going to miss some compounds. Typically, what you would have is an activity distribution like the one shown on the right, with many compounds showing really no activity on your target, and the higher the activity, the less number of compounds you will find. Last but not least, this pilot phase can be used to learn about potential interference mechanism to fine-tune your hit validation strategy, and we'll go into that in the next webinar. So now you're ready to run your screen. Your compounds are plated in 96, 384, or 1536 well plates. Some of the wells contain the positive control, some may contain negative controls, and DMSO as quality control, DMSO often being used as the actual negative control. Typically, you screen your compounds in concentrations ranging from 10 to 30 micromolar. As a result of your screen, you may get a heat map like the one shown on the right side of this slide, where you see on the far right, the column is filled with positive controls, which you use to monitor the assay performance over time. And then the colors correspond to individual compounds. The blue are those showing inhibition, the white no effect, and the red activation. And the stronger the effect, the stronger the color. Another way to plot the data is shown on the bottom left, where each dot is a particular compound. And what you can do here is, based on the activity in the y-axis, select the range of activity that you want to pursue as hits. So you will select a cutoff, usually based on the hit rate and the capacity of your validation assays. Typically, you want a hit rate below 1%. What you usually want to do is to retest the hits in the same assay and then select only the ones that confirm. And the next step is to run a concentration response curve where you would test different concentration of your compound. And here it's important to not only look at the numerical values, the IC50 for example, but also the shape of the curve. Typically what you want to see is a sigmoidal curve like the one in blue, but other compounds may lead to different types of curves, such as the one on the right. And there should be a flag, not necessarily meaning that you should discard these compounds, but certainly that they should be looked at in more details during the validation process. So really important, numbers can hide the reality. Look at the curves together with your screening expert and biologist. This is, to finish, an example of an unbiased screen. In this case, the target is SPPL2A, signal peptide peptidase-like 2A, which is an interesting target for autoimmune diseases. In this case, the researchers went for the full screening set, 1.4 million compounds. The assays allowed it, and they thought they needed that in order to find interesting starting points. The assay utilized is a high-content imaging assay using cells expressing the target, SPPL2A, and a fluorescently labeled substrate, EGFP labeled TNF-alpha, which is a peptide that is expressed in the membrane. When the substrate is cleaved by the enzyme, the fragment containing the fluorescent label will be translocated from the cytoplasm into the nucleus. So you can monitor the inhibition of SPPL2A by looking at the ratio between nuclear and cytoplasmic EGFP intensity. The assay setup was 1536 plates because of the large number of compounds, 20 micromolar test concentration, using DMSO as a negative control, and a known inhibitor as a positive control. And you see that Z prime was excellent. Now, what's interesting is in this example is that when they looked at the hits that showed greater than 40% inhibition, they found 93,000 hits. This is a very large number, more than 6% hit rate. And they also saw, from first inspection, many false positives. Now, the beauty of using high-content imaging is that you can look at many parameters in the cell. So what they did is multi-parametric image analysis to try to see compounds that has similar effects compared to the positive control. And with this, they were able to narrow it down to 39,000, so more than half remaining hits, and a much lower false positive rate. We come now to the second approach, the focus screen. And here the objective is to use the knowledge we have on the target to select a set of compounds for screening, which we believe will have a higher chance of activity. So there are three approaches to do that, and in each case we can either select from existing screening sets or make a dedicated library. So these can be compounds with activity on a target class. So let's say you're working on an ion channel or a kinase. You can select compounds that have activity on other targets in this family, and these will have a higher chance of being active on your target of interest. The second approach is to use compounds with a desired mode of action. So for example, if you want to go after a target with covalent inhibitors, then you can select a set of compounds with reactive groups. And finally, you can select compounds with structural motifs which are known to interact with your target. So there are three examples below. On the left, you have kinase hinge binding motifs, and many of the kinase inhibitors contain one of those, and they simply have a hydrogen bond donor and acceptor. In the middle, if you're working on a metalloenzyme, then you can use compounds containing metal chelating groups such as carboxylates, hydroxamic acid, thiols, etc. The example on the right is a little bit more elaborated. If you're working on a serine protease like trypsin, you can use what is known to be the requirement for the compound to bind in the very important S1 pocket, which is a positive charge to interact with an aspartate, and a certain size so that it can enter the S1 pocket. So this is an example of a focus screen where the researchers were looking for inhibitors of a mutant EGFR kinase, but importantly with selectivity over the wild-type enzyme for cancer. In this case, they started by selecting a 20,000 diverse kinase reversible inhibitor targeted library. So 20,000 kinase inhibitors, which they screened using an enzymatic assay looking at product formation, ADP, and they had both a wild-type assay and an assay using double mutyl kinase. They found one compound, which is shown on the upper right, which shows the appropriate level of selectivity as well as high potency. Now, one challenge that they faced is that the reversible mode of action of this compound was leading to poor cellular potency. And the way they address this is by adding a covalent warhead with this acrylamide to improve cellular potency. And this they did using a binding model to place the acrylamide so that it could react with cysteine 797 as seen in the bottom picture. They could further elaborate this compound into the drug candidate, which is shown at the bottom. So I think this is a really nice story because only by screening 20,000 compounds, they could actually find a hit that they could turn into a drug candidate. And it shows you that for targets where we have a lot of knowledge, and kinases are of course a great example, we can actually, by just screening a few compounds, find very interesting starting points. So the last of the classical screening approach is the in silico based screening. So here we're going to use the target knowledge to pre-screen in silico using computational methods and do that to reduce the number of compounds to screen experimentally. So if you would run an unbiased screen in HTS, you would typically have a hit rate around 0.1%, maybe a bit lower or higher. But if you enrich using in silico screening, by combining in silico and experimental screen, you can get a hit rate 10 to 1,000-fold higher. An additional opportunity is that you can expand the compound set beyond the one that you actually have in-house. You can screen commercial compounds or even compounds that don't exist, virtual compounds. There is, for example, the real space, which contains 14 billion make-on-demand compounds that you can purchase if there are hits from your in silico screen. So we're going to go into three common approaches: ligand-based, structure-based, and statistical model. Starting with the ligand-based, here we start from a known chemical matter, a compound or several compounds, and then we're going to mine a compound collection for compounds which have a chance to bind to the target of interest. We can do that using similarity based on the chemical structure. This is the left part of the slide. The hypothesis being is that similar compounds have similar biological properties. So most commonly, we would simply look at the 2D fingerprints, so the 2D structure, but you can do more elaborated approaches looking at 3D shape or molecular fields. So the cartoon at the bottom is trying to illustrate how this can look like. You have inactive compounds in red, you have active compounds in green, and you cluster similar compounds to these in several clusters, and you will end up with clusters of compounds which are predicted to be inactive and clusters of compounds which are predicted active. The idea is that if you only screen the clusters which are predicted active, you have a higher chance to find hits. On the right side of the slide, you have a second approach, which is called pharmacophore-based. Here, we take the hypothesis that the essential interactions between ligands and target and their 3D orientation can be conserved among several compounds. So, if you think about a structure like the one in gray, you can reproduce it according to its pharmacophore. So all the polar groups can be either hydrogen bond acceptors, hydrogen bond donors, cationic, and ionic. And then you would have hydrophobic groups and aromatic groups. Then you get the picture on the right, where you have the interaction types and where they stand geometrically. And the concept is that if you find compounds which have similar interaction types and in similar geometries, these have a higher chance of being active against your target. There are also other ways to do ligand-based selections, like activity-based fingerprints, which uses rather biological data and not chemical structure. The second type of in silico screening approach uses structure-based or docking approaches. So here we're using the structure of the protein that we get either by X-ray, NMR, or homology model to predict if a molecule will fit in the binding site and make favorable interactions. So you have an example at the bottom. You start with the structure of the compound, the 3D structure. Then you have the structure of the protein. And using computational methods, docking methods, you will place your compound inside the binding site and determine possible conformations of the ligand in the protein. In the end, you will also select the most likely, lowest energy conformation of the ligand. Now, importantly, this is always followed by a step of manual inspection where you verify that what the computer has produced makes sense from an interaction and geometry point of view. Now, this is a very powerful approach, but one has to consider a couple of limitations. First, it's important to have high-quality models, whether they're from homology models or from an X-ray structure. It's also important to consider the flexibility of the binding site. The structure gives you a very static view of the structure of the protein, and obviously not everything can be predicted. The ligand can have consequences to the structure of the protein and lead to movements of some of the residues. The last approach for in silico screening is using statistical models. And here we use the activity data from a large set of known active and inactive ligands to predict the likelihood that a compound is active on a target. So a good way to explain this is to look at an example. So here, the example is called Profile QSAR for Quantitative Structure Activity Relationship. And here it starts with a lot of data. We have 18 million PIC50, so 18 million activity data points for many compounds in many assays. Now, the data we have is illustrated on the upper right part of the cartoon. We have on the horizontal axis the assays, and on the vertical axis the compounds. And you see we have a lot of data, but of course not all of the data. And if we use this as a starting point to predict the activity of these compounds in a new assay, this would not lead to very successful results. This method uses a two-step process. First, we fill the missing data points by building single assay QSAR models, which gives us the picture at the bottom where we have a completely filled data table for all assays and all compounds, some of which are experimental, some of which are predicted. And in the second step, we use this predicted activity profile as compound descriptors to predict how the compounds will perform in the new assay. This has been quite successful for target classes with many data points. Kinases is probably one of the most prominent examples. But more recently, it was shown that it can also be successful in phenotypic approaches. This is an example of an Insidical screen. Here, the authors were trying to find cGAS inhibitors, cyclic GMP-AMP synthase inhibitors, which is the target involved in autoimmune diseases. They started with 1.5 million commercial compounds and used a docking approach. They had a next-ray structure of the protein, and more interestingly, a protein complex with an inhibitor, as shown on the right. After clustering and visual inspection of the docking scores, they could select 59 compounds that they purchased for testing using a thermal shields assay and a biochemical assay, which led them to 4 hits, one of which is illustrated on the bottom right part of the slide. So I think this is a nice example because by only testing 59 compounds experimentally, they could actually find hits. And this shows you a little bit how powerful this docking approach can be when it's successful. So this brings us to the end of this webinar. We've looked at 3 different classical screening approaches: the unbiased screen, the focus screen, and the in silico based screen. And they differ essentially in the chemical space we screen. The unbiased screen and the focus screen will look at existing compounds, whereas the in silico based screen can look at both existing and virtual compounds. The number of compounds can also be very different. Usually the unbiased screen looks at a number of compounds that depends on the assay throughput and costs, but usually it can be quite large, from 10,000 to several millions. The focus screen uses a reduced set based on existing target knowledge, and here we're usually in the range of 50,000. The in silico based screen can also be quite small, but can go to several millions depending on the computational method. The advantage of this method is, of course, that we only have a reduced set of compounds to test experimentally. Obviously, they all come with opportunities and challenges. For the unbiased screen, the big advantage is that because you're unbiased, because you start with a larger set of compounds, you can identify diverse hit series covering multiple binding sites and modes of actions. On the other hand, it comes usually with a high cost, and it's time consuming, both for the screen itself, but also for the validation process. And finally, because there is intrinsic variability in the assay, you have a risk of missing weak hits in the noise. The focus screen is usually faster and cheaper because we don't screen as many compounds. It has a high chance of success, and you can use complex assays. But the challenge is that you restrict yourself in terms of diversity, and you're limiting yourself to known binding sites and MOAs. The in silico screen probably covers potentially the largest chemical space. You can also use complex assays because in the end you only screen a fraction experimentally. But it comes at another cost because you need additional computational resources and high quality models. In this case, we usually also restrict ourselves to known binding sites and MOAs because we start from the knowledge we have. With this, we come to the end of this webinar. Please don't hesitate to leave us comments, ask questions. We'll be happy to get back to you.